Namaskar dosto, welcome to channel Toxicology on Board. Today in this video we'll be discussing another important topic with respect to lead. As I told these metals, whether it is lead or arsenic or mercury, there are certain important metals which are ubiquitous in nature, where human as well as environment or soil, plant, material, minerals will easily get exposed to this particular metals and which will cause toxicity. So these questions, these type of metals, they are the important questions for this DABT examiner. So the last video also we have learnt about lead with respect to elimination, route of elimination. What is the primary route of elimination for lead? So today, same with respect to lead only, we have come up with another topic, that is the intranuclear inclusion in the renal tubular epithelial cells. Okay, if you have to understand that last video, last question was also taken from ABT recertification examination May 2000. Okay, so now you can understand within the same year, same question paper on the same concept, that is on the same topic, two different questions have come. So certain times this will happen on the same topic only. You can get more than one question, I mean to say. So let's see what is the important question that is given with respect to lead. The question was intranuclear inclusion in the renal tubular epithelial cells is the characteristic of okay, so this is we have to remember with respect to histopathology. Once a person is exposed to lead and that particular kidney if you take and you are applying this histopathology, what are the classical features with respect to this pathology of this particular kidney and which section within the kidney also you can see there are different parts are there: proximal convoluted tubule, tubular loop of Henle, ascending descending nephron, glomerulus. In that also particularly it is affecting PCT. Actually the major route of excretion for the lead is urine. In that also you have seen that PCT is the thing where inclusion bodies are there. Previous video if you see, in that also some part of the lead is tubularly it getting reabsorbed. So let's see what are the question, what is the option. So intranuclear inclusions in the renal tubular epithelial cell is the characteristic of option A is cobalt, option B is mercury, option C is antimony, option D is lead, option E is cadmium. The right answer will be in the green text, that is option D, lead. Okay, so the characteristic feature of lead is intranuclear inclusions in the renal tubular epithelial cells. PCT may this will be there. So let us understand what is this intranuclear inclusion. Intranuclear inclusion in renal tubular cells. Here you can see these tubular cells will be there, no, are abnormal proteins. Okay, lead will form, lead protein complex will be formed, which will be there in the renal tubular cells, or viral collections within the cell nucleus. Okay, one more picture also I will show you, often seen in toxic like lead poisoning. So it is a characteristic feature of lead poisoning. Also it is seen in the case of viral infections. What are those viral infections? Polyomavirus or drug induced kidney injury. There are certain drugs which are also causing this kind of same characteristic feature, that is DIKI, drug induced kidney injury, particularly chemotherapeutic agents, and can appear as dense eosinophilic bodies on microscopy. When you see under microscope this histopathology slides of this particular tubular section, no, here you can see these arrow marks are given, no, those are nothing but your dense eosinophilic bodies on microscopy, indicating cellular stress or pathology. So the cell has undergone lot of stress because of the exposure, excessive exposure to this lead. So this is the particular classical pathological feature. Though sometimes they are idiopathic, sometimes we cannot identify the cause for this particular change, and less harmful, especially in the research animals, highlighting the need for careful diagnosis. So let's understand more in detail more. Here you can see the red or pink color deposition in the cells you can see. Let's understand in detail. So what are the common causes? As we have seen, either it is caused because of this particular metal, that is lead toxicity. It's a classical cause, forming dense dark staining inclusions in the PCT. Here you can see, right, as a protective mechanism against lead. One you can understand. Okay, undergoing stress cells, PCT cells are undergoing stress. To as a protective mechanism, this kind of characteristic feature is developed. Viral infections, what are those are there? DNA viruses like adenovirus or cytomegalovirus CMV and polyomavirus can cause inclusions often with the chromatin margination. Next, what are the under DIKA? DIKI, drug-induced kidney injury. Certain medications, including some chemotherapeutic agents and toxins, can induce these inclusions, which may be proteinaceous. Okay, viral protein will be formed with respect to lead. Then these kind of deposition or inclusion bodies can be formed in the PCT. Okay, then as I told you, another thing is idiopathic or spontaneous. We don't know the reason. Once we don't know the cause, then it will be very challenging for the physician to treat this particular thing. In some cases, particularly in immunodeficient mice, these may be spontaneous degenerative changes without a detectable pathogen. So in the case of research animals, that is immunodeficient mice, this particular thing has been observed. Okay, so what are the characteristic features of this inclusion bodies is that appearance often eosinophilic pink or red. As I told you, this you can see this arrow marker, and dense on light microscopy, varying in size and shape. Certain places it is very small, certain places it is very big. And once you observe the same slide under electron microscopy, it will show granular and fibrillar material with displaced peripheral chromatin. Okay, and as I told you, which part of the kidney it is affecting primarily in the proximal renal tubular cells, that you have to remember. Then next we'll see what are the important clinical significance by identifying this particular inclusion bodies. How it is benefiting to the clinician or physician is it is an indicator of injury. As I told you, early response signifies cellular stress or damage, but the severity varies from the individual to individual, exposure to exposure. Coming to second one is diagnostic challenge. If we know the cause, whether it is because of the lead or because of the virus or because of the particular drug induced KI, then accordingly we can treat that particular patient. But if it is idiopathic and we don't know the cause, then it becomes a challenging for the the physician to treat it. So diagnostic challenge, distinguish between toxic, viral and idiopathic causes is crucial for the patient management. Coming to monitoring, it can be exfoliated into the urine, okay, serving as a marker for exposure, lead or disease. Okay, if it is exfoliated in the urine, you can perform urine analysis and it can be used as a marker for this exposure to this lead toxicity. Then next is we'll see, so what are the important things that we can get it from this particular finding is that finding this intranuclear inclusions in the kidney cells is a significant histopathological finding. Okay, pathologist will be reading these slides, requiring correlation with patient history. Then you have to check, okay, there is a question and answer should be given to the patient history, and further testing viral PCR and special stains to determine the underlying the cause, which can range from treatable toxicity to viral disease or even benign spontaneous changes in certain contexts. So here you can see the pink or red color, this particular inclusion bodies, you can see the arrow mark, and this is a normal cell. This is the outermost layer is the cytoplasm, and in this is the nucleus, and here you can see cells with intranuclear inclusion here. This is the normal cells and this is the cells with the intranuclear inclusions. Hope from this video where you were able to learn about this lead, particularly lead toxicity, particularly its histopathology and a classical feature, intranuclear inclusion. Okay, along with this video, also see my previously uploaded video. In that also I mentioned about this inclusion bodies and particularly PCT, percussion convoluted tubules, how it is affecting. Okay, thank you so much. Be curious. For any questions, please write a mail to arya.priyanka18@gmail.com. Like, subscribe and follow my YouTube channel, Toxicology on Board. Thank you so much. All the best for your exam.